Hello and welcome to the QPS webinar on excellence in complex clinical trials. We are beyond thrilled to have you here, and we will share about 3 case studies with you in just a minute. Before we get started, I just want to give a little bit of a background to who QPS is. We are a clinical research organization that delivers the highest grade discovery, preclinical, clinical drug development services. We've been in business since 1995. We started as a small bioanalysis shop back in 95 and have now grown to a full service CRO with over 1,200 employees across the globe between Europe, Asia, and the US. We have expanded pharmaceutical contract R&D services with expertise in neuropharmacology, in DMPK, toxicology, bioanalysis, translational medicine, asset development strategy, and early to late phase clinical study conduct. We're known for our ability to be agile and flexible and work with our clients across the development lifecycle. With that, we are thrilled to jump into our webinar. Just a couple of housekeeping points for this webinar. It's going to be recorded. You'll get an email about the recording after the webinar where you can go back and watch the on-demand link at your leisure. Also note that there is an option to submit questions in the Q&A box. We'll answer as many of those questions at the very end of the webinar for all 3 of the case studies that we'll review. If there's a question we don't get to, don't worry, we'll circle back on that and send an email with the appropriate response. With that, we'll move to the next slide, and I'd like to just introduce myself first. I'm the Executive Director and Head of Global Medical Affairs at QPS. My name is Sarah Cross. With me, I have 3 incredible scientists, Dr. Mahrika Brill, who is our Associate Director of Clinical Pharmacology. She will be taking us through a case study on biomarker-driven bone marrow assessments. Our second case study will be by Dr. Isaac Dendas, who's our Director of Clinical Pharmacology and Patient Services. He will be taking us through a case study on full sedation. And our last case study will be led by Dr. Peter Dogterom. He's another Director of Clinical Pharmacology at QPS, and he'll be taking us through an LPS challenge case study. So without further ado, I'll turn it over to Mahrika.
Thanks a lot, Sarah. So hello, everyone. Today I'll present a case study that about a study where we collected bone marrow cells in healthy volunteers to inform on the cell cycle status of bone marrow cells. And it was a clinical pharmacology study which we performed in healthy volunteers. So a brief overview of this study. It was about a potentially chemoprotective agent. As I mentioned, the study was performed in healthy volunteers, and the main objective of the study was to identify a dose at which the IMP showed preferential cell cycle rest in bone marrow cells. So very roughly, the study was set up as follows. You see on the left-hand side, we get 1 dose was given, and then the safety, tolerability, and all endpoints, mainly PK and safety, were reviewed by a safety review committee, which then decided on whether to escalate the dose or repeat or go to a lower dose. And then the second dose was given, and then this process was repeated until all the doses that was aimed for at testing were performed. So there were quite a lot of endpoints in the study, which is one of the reasons it was quite complex. And the main endpoint was the flow cytometry of the bone marrow, providing information on the cell cycle of the bone marrow cells. In addition, a lot of biomarkers in plasma or peripheral blood mononuclear cells were collected, the skin biopsy was performed, and also pharmacokinetic information in plasma and safety and tolerability were collected. So a rough outline of the study is as follows. Patients entered the clinic on day -1 to recheck eligibility for the study, and then on day 1 they received IMP infusion, and of course before that all the pre-dose assessments were performed, and the colors of the arrows correspond to the endpoints on the right-hand side of the graph. So after dose, there were PK, PD collections in plasma, safety endpoints, and then exactly 12 hours post-infusion, which coincided with the morning of day 2, the bone marrow biopsy was done to collect bone marrow cells and a bone marrow biopsy. In addition, also at that time point, a skin biopsy was performed, and basically all of the other assessments, so PK, safety endpoints, PD endpoints in plasma, etc. If all was fine with the subject, he would return home on the same day, typically in the evening after the collection of the last assessments, and then there will be a safety follow-up on day 7 and day 28. So when looking at the study, there were mainly 3 aspects which contributed to the success of the study, and those are listed here on this slide. First of all, of course, this study required a bone marrow biopsy and aspiration, and that's quite a specialized procedure for which you also need a lot of routine practice. So it's not something the QPS PIs are doing on a daily basis. The second, I could say challenge or complex part of this study was the work of those bone marrow aspiration, so the bone marrow cells that were collected. Those should be kept fresh, they could not be freeze down, and should be worked up within 4 hours after collection, and actually the flow cytometry to inform on the cell cycle should occur the same day. Then lastly, of course, the recruitment of healthy volunteers really how that will contribute to the success of the study. So could we do this in healthy volunteers? Is it safe to collect bone marrow samples in these patients? So when looking at these 3 aspects, mainly the local collaboration and the subject local collaboration that we have here in Groningen, and the subject engagement to the phase 1 clinic that we have here really contributed to the success or to the conclusion of the study. So for the bone marrow biopsy and aspiration, we collaborated with the hematology department at the University Medical Center in Groningen, and this is a large university hospital, and the QPS clinic is actually located on the premises of this hospital. We have been partnering with UMCG for many studies and with different departments, and for this one we were having a partnership with experienced hematologists that could that actually do these bone marrow biopsy and aspirations on a daily basis, and could also help us with this health volunteer study. So then if we come to the second thing, the workup of those bone marrow aspirates within 4 hours and doing the flow cytometry the same day, the QPS, besides the QPS clinic in Groningen, we also have a QPS lab, bioanalytical lab, very close to us. It's a 5-minute walk. And this QPS lab also has a small flow cytometry team, which was able to support this study and validating the methods and supporting. And actually, the team is really flexible. They could even be at the clinic. So immediately at the time the bone marrow aspiration was collected, they could immediately put it on ice and transport it to the lab to start the workup of the bone marrow aspirate and do the flow cytometry on the same day. So these two collaborations were of course great. So that was of course one step, but then also really bringing together the study really required very strong planning, strong communication, and cooperation from all the stakeholders involved. Then lastly, the recruitment of the healthy volunteers. We could do a safety assessment to see, is it safe to dose IMP in this type of population? Is it possible to do bone marrow biopsies in healthy volunteers? Well, in collaboration with the hematology departments, it seems that it was totally possible to do this. And then actually the recruitment of healthy volunteers turned out to go very smoothly without a lot of hurdles. And when looking back I think one very important point is the communication towards the potential volunteers to volunteer for the study, being really detailed about what procedures they could expect. First local anesthetic, then the actual aspiration, which could hurt at certain moments. And I think in the end, the high trust in the QPS researchers and study personnel, as we've seen in many surveys over many studies, really contributed to volunteers being willing to participate. And yeah, actually that going very smoothly. In the end, actually for people that did participate in the study, a small survey was done, and actually 9 out of the 10 subjects that had participated in this particular study mentioned that they would do this again with a future study, having a similar assessments. So all in all, these aspects, having a single location with our phase 1 clinic, having the flow cytometry supports of our bioanalytical lab, and then having the expert hematologist engagement for the study really brought everything together. And in particular for the study, this allowed the data coming from the study, allowed the sponsor to establish a dose response curve on cell cycle arrest and in human bone marrow cells, and thus provide them a very rapid insight into the mechanism of action and human pharmacology, which they of course required for planning their ongoing phase 2 studies. So with this case in mind, of course, we can think about other scenarios having these aspects in the study. We could, for example, think about studies where we do 2 bone marrow biopsies pre and post dose. We could study duration of effects on bone marrow cells, or could combine with PET imaging that we've done actually for other QPS studies. I think there's a lot we can think of, and we're happy to discuss your thoughts and questions about this. So thank you very much. I'll bring it over to Sarah.
Fantastic. Thank you, Margarete, for that nice overview. With that, we'll move on to Dr. Dendas to take us through our sedation in healthy volunteer study at QPS.
Thank you, Sarah. This is a clinical pharmacology study in healthy volunteers, full sedation study, performed at the clinic at QPS, which is actually on the premises of the University Medical Center in Groningen. QPS initiated a range of sedation studies in early 2014 for the company called Innovation. All the data I'm going to show you right now has been published, so we can easily talk about compounds and results here. ABP 700 is a potent positive allosteric modulator of the GABA-A receptor, and is an IV anesthetic being developed for monitored anesthesia care. The target was patients undergoing diagnostic or therapeutic procedures and or general anesthesia in patients undergoing their diagnostic or therapeutic procedures. The background of ABP 700, it's a second generation soft analog of etomidate. It has a ester linkage, which gives rapid induction and emergence of anesthesia, and the major metabolite is inactive. And in the preclinical studies, the results were encouraging. You can see that the half-life is relatively low, 5 to 12 minutes. This compound is developed in the labs of Dr. Rains. He's a professor of anesthesiology of Johns Hopkins Hospital. On the right, you see through a window of the room where we performed this, where we performed the whole range of studies with a team of anesthesiologists and nurses and all other co-workers for this study. The study design and cohort overview is as follows. It was ethics committee approved. It's a randomized double-blind placebo-controlled studies, single ascending IV doses, ranging from 0.03 to 1 mg per kg. The healthy volunteers at ages between 18 and 45, we started in the first cohorts with only male volunteers, but I think from cohort 5 we started with female volunteers as well. Totally 60 subjects, 5 active and 1 placebo at every dose level. The study setup was a fully equipped room as an OR. It was actually an OR we designed on our premises, of course, without the surgeon. There was full capnography. The bispectral index, the BIS equipment was used. It is an EEG apparatus of the frontal cortex, and you can see the readout on the above right photo. ECG, vital signs, full and real-time data capturing called the RUC loop, especially designed by the anesthesiologist department of the University Medical Center Groningen, which means that all the data of all the parameters could actively go into a database. There was venous and arterial sampling. Of course, there was a crash cart in case of emergency, and also blood gas analysis. On the second photo, you see the blood gas analysis on the right, on the left photo, on the left of the photo. We could do up to 4 subjects per day, dosing group every week, which meant actually that this was a very fast throughput. There was a big dedicated team consisting of an experienced anesthesiologist, an anesthesiology assistant, data capture assistant, coordinator for the arterial sampling, venous sampling nurse, and sample processing nurse. The results here, first of course, safety, tolerability, and PK, and we've got the PK here. The safety and tolerability of all dose groups were good. And here's the PK with times in minutes. And you can see a good dose-dependent increase of ABP 700 levels with increasing dose. One of the disadvantages of etomidate, which is an anesthetic widely used up to the 70s, perhaps even up to the 80s, is that there was adrenal suppression. So we did during our dosing also an ACTH challenge of 250 micrograms, and you can see that there is no clinically significant adrenal suppression in all dose groups. So the disadvantage of etomidate you could not see with ABP 700. Another important thing is, of course, the sedation. And for the sedation, there were several tests. One was the MOAS test score, which is calling the patient if he's still awake, and also some manual tactile doing of the patient, so asking him, are you awake, and then putting your arms in his neck, etc. But a more objective parameter is the BIS changes, and that is the frontal cortex activity. And if you look at the placebo at 0 mg per kg, it's a relatively straight line, and you can see, funnily enough, that later in the time, that BIS score goes down because the patient is lying there for half an hour and gets dozy and starts sleeping. But the more important thing is, of course, that sedation kicks in at the 3rd dose. It begins pretty well, but real sedation you can find on average with a BIS of, let's say, 60% of 100, and you can see that serious sedation starts with approximately 0.35 mg per kg. But it's also a short-lasting sedation, so and only takes no more than 15 minutes. So for little acute operations, this would be an ideal candidate. So we can conclude from the pharmacodynamics that there are no SAEs, there were no significant laboratory findings, no adrenal suppression, no pain on injection, and low incidence of nausea and vomiting, which is with the competitor like propofol, where there is often complaints of pain on injection and an advantage. There was some involuntary muscle movement at the larger doses of 0.175 mg/kg. Short-lasting periods of apnea, but not serious. Dose-dependent increase in heart rate, which was actually a good sign, because loads of anesthetic give a decrease in heart rate, which is undesirable, and blood pressure. And the stopping criteria reads at 1 mg/kg. So overall, linear kinetics, fast onset and offset, anesthesia from 0.25 mg/kg, well tolerated up to 0.75 mg/kg, and safe to proceed to the next level, which would be a whole set of infusion studies. So the takeaway message is that you can perform a study like this at the CRO, at their clinics, but this is only possible in close collaboration with academic KOLs, anesthesiologists, and of course a CRO which is experienced and flexible as QPS is. Thank you. I'm going now to the next person.
Wonderful, thank you, Isaac. With that, we'll turn it over for our final case study with Peter to take us through our LPS challenge study.
Yes, hello everyone. Nice to meet you, and I'm happy to present you in this row of presentation the final study to be shared with you, and that is called an LPS challenge study. LPS challenge study, what is LPS doing? LPS is known to induce the production of pro-inflammatory cytokines, and that's a kind of model for inflammation as a reflection of quite a number of therapeutic indications. So in itself, it's a nice tool to apply in your early phases when you have a compound of interest which is acting via that pathway. But before you start this LPS challenge study, first you have to overcome other challenges. And here I listed a couple of them. First of all, you need to obtain GMP LPS. GMP because you're working with healthy volunteers, so you have to follow the GMP rules regarding what you administer to human beings. As already mentioned in the previous presentations, we are QPS Clinics is on the premises of the University Medical Center Groningen, so a big source of specialist KOLs. And also for the LPS challenge test, we work together with intensivists from the UMCG, very helpful, very experienced, so a great contribution to the preparation and the execution of this trial. We have to realize it's not a high risk, but you expect adverse events. We come to that later. You can only dose a restricted number of volunteers per day. So we can think about, let's say, 2 to 4 subjects as a max on a single day. You have to inform the volunteers on expected adverse events. It's known LPS will induce transient flu-like symptoms. Well, in itself, they are not, let's say, severe or of course not life-threatening. It causes discomfort, and people can feel, let's say, having a real flu. But as long as you inform the volunteers upfront in a very transparent way, this is what you expect, what we expect. They know, well, when they experience these effects, it is as expected. So that will recover. And it is transient. So in most cases, 24 hours after the onset, it disappeared, and they are as healthy as prior to the start of the study. In order to safeguard the volunteers, you have to record vital signs, ECG and oxygen saturation. And in case of you need to be sure that you have available symptomatic treatment in case there is a medical need. In addition to that, it's very helpful when you have, let's say, a very experienced IRB. And experience means very familiar with complex phase 1 studies, like the studies discussed before. So an IRB, which has a good understanding what's going on in clinical pharmacology in the early phase, they can understand where you come from, what the goal is, what the risks are. They know QPS, so that they really can judge whether this is ethically justified or not. So that is very helpful. Of course, you have to discuss with the sponsor the design of the study, because there are various ways to run an LPS challenge study. You can do it in a parallel setting, you can do it in a crossover setting, you can do it upon a single dose of the compound of interest, you can do it after multiple doses. So there are a lot of things you have to realize and to discuss with the sponsor, and also it will be very helpful if the sponsor already has preclinical information or done these type of studies preclinically in mice or rats to build comfort that this might work in healthy subjects as well. So the study I would like to share with you was a first-in-man study with an IRAK1, IRAK4 inhibitor. So the sponsor conducted already preclinical research with LPS and showed positive results in this setting. But before you do the LPS challenge study, you need to collect information on the single dose safety, tolerability, pharmacokinetic in an ascending way, as well as the multiple dose safety, tolerability, and pharmacokinetics before you embark on the LPS part. So SAD, MAD, and LPS is part of what we call an umbrella protocol. So you present it as a package to the IRB, so they understand where you come from, what the goal is, and also in the context of. So that is very helpful rather to do it as a standalone study. So the main objective of this LPS part was to characterize the PD profile of the compound of interest following an LPS challenge in healthy subjects. For the LPS part, we had 16 healthy volunteers, 8 on active and 8 on placebo in a parallel design. So schematically, you see that the SAD part consisted of 8 ascending dose levels. The MAD part consisted of 2 dose levels administered for 14 days. And then thereafter, the dose was selected for the LPS study. And that was a multiple dose, and upon the final dose, the LPS bolus at steady state. The endpoints are cytokines. You can analyze a bunch of cytokines, but for this particular study, we collected serum for CRP, TNF alpha, IL-6, IL-8, MIP-1 alpha, and MIP-1 beta. And the purpose was to do frequent blood sampling in order to construct a time concentration curve, which allows you to calculate the AUC and the Cmax and the Tmax. So zooming in on the LPS design. So the design and the dose selected for the LPS part was based on the collected PK and safety profile in the SAD and the MAD parts. The profile of the drug was great, no safety tolerability issues. The pharmacokinetics showed that the Tmax was around 2 hours post-dosing. Steady state was obtained after 3 days of BID dosing. The half-life was approximately 4 to 7 hours. So we decided to have the IV bolus of LPS at the Tmax of the compound, so that is 2 hours post-dose, and the dose of the LPS was 2 nanogram per kilogram. So that is schematically presented at the bottom of the slide on day 4. So the dosing of compound of interest and the bolus thereafter, and then 48 hours thereafter, when all adverse events disappeared and the subject was back to, let's say, a healthy state, was discharged from the clinic, and just one follow-up on day 13 to make sure that there were no residual effects and that the subject could be released from the study. As mentioned, the endpoint on day 4, safety tolerability, and then the PK over 50 hours and the PD over 50 hours. So the results, so let's first zoom in on the safety and the tolerability. All subjects reported at least one treatment emergent adverse event. So let's say clustered as an influenza-like symptom. So you can think about headache, malaise, feeling uncomfortable. So the typically flu-like symptoms related to LPS. And basically, there was no difference between the active and the placebo cohorts in terms of these LPS-induced side effect. Half of the adverse events were of moderate intensity, but all transient. The rest were mild, had a mild intensity. And as expected, transient increases in body temperature, blood pressure, pulse rate, but there were no clinically significant abnormalities, so no need for any intervention. Overall, the safety and tolerability profile upon LPS challenge was in line with what has been reported in the public domain. Some results regarding the cytokine responses. So you see the dotted lines are the placebo curves, and the solid lines are on top of the compound of interest. And the numbers you see is the reduction at that time point. So when you look to TNF alpha, you see that the maximum reduction was 78%. But what you see in general is that without the compound of interest, the expected increases, transient increases of the various cytokines, which disappeared depending on the cytokine more rapidly or more slowly, but all were expected and shown here in this graph. And what you also see is that the compound of interest had positive effects on the cytokines. So there's a clear reduction in the induced cytokine response. CRP is the only one where there is no difference, but likely that has to do with the rather slow response of CRP upon an LPS challenge. That was not a surprise. That was in line with what literature said. So in order to investigate that in more detail, you had to play around with the time of dosing. But in this study, we only had one time point of dosing the LPS. So the conclusions are that this study was able to provide rapid insight into the PD of this new IRAK1, IRAK4 inhibitor. Rapid insight means, okay, with one design, we have demonstrated that there is a pharmacological effect. So as the compound works as it was expected to work, and the safety and tolerability profile was in line with literature, so that there were no surprises regarding safety and tolerability. The take-home message is, and that is in common with what my colleagues already presented for their studies, is that you need to have a recognized team of experts, and we are in the fortunate position that we are on the premises of the UMCG, the largest academic hospital here in the Netherlands, and there is a great interest of many of these specialists to work together with with us to be involved in innovative drug research. So from a scientific point of view, they are interested, but also from a medical point of view, because these are typically the new compounds of the future. And the risk of an LPS study is similar to other complex phase 1 studies. QPS here in Groningen is well known that we are able to run complex phase 1 studies. So we have a lot of assessments, we have a lot of innovative methodologies. So there is a kind of risk, but we have a great team to prepare these studies and to make sure that what we are doing is under well-controlled conditions with a very experienced medical team, a scientific team, and a medical environment is located at the UMCG. So with that, this was the final study we would like to present and share with you. So thanks for your attention, and I handle it over to Sarah.
Wonderful. Thank you, Peter, for that for informative overview. And thank you to all three of our QPS experts for taking us through those 3 case studies. I will say we had a couple of questions come in as you were speaking. Peter, why don't we start with you first?
That's fine.
The first question we had, which is, why would you need an extra LPS study rather than immediately testing efficacy in patients?
Yeah, that's a great question, a well-known question. I can remember in the early days of biomarker research that there were companies who said, okay, we plan to go as fast as possible in patients. Others were more forced in biomarker. It can help you, especially what I mentioned in my presentation, when you incorporate it in your umbrella protocol, it doesn't take too much extra time. So be aware on that. You can have a rapid readout when you build it in an existing protocol. Of course, you can do it as a standalone, but that costs more time and more cost. So nowadays, when we have, let's say, validated biomarkers coming from the preclinical research, and there is based on other research, a correlation with, let's say, the medical outcome, it can definitely help you to build your comfort, because that's basically what you are doing during your phase 1 program, build comfort in your compound. So it is an extra stimulus to say, okay, it worked pharmacologically, and even it can help you in the dose selection for your next phase.
Very informative, Peter. I think the second question that I would ask is, then why would you execute the LPS test at a steady state compound and not after a single acute dose?
Yeah, well, what you see is that basically, of course, you can fine tune that. You say, okay, let's do a single dose, let's do it multiple dose, let's administer the LPS at various time points, but then you make it maybe at this stage more complex than really needed. I think the most important thing is that you would like to have, let's say, proof of concept of your pharmacology, and therefore take let's say the worst case scenario. It can be that your molecule has to induce certain systems which are more active upon multiple dosing rather than on single dose. So it is also kind of risk. So I think this is the most safest one, that you really have steady state, a concentration which you have, expected concentration you have derived from your preclinical work and look into the future. So keep it simple, do it in the right way. And if the outcome is very instant, there's always time for fine tuning. But at this stage, do it in this way.
Wonderful. Thank you, Peter. Really appreciate the insight there. And again, thank you for the wonderful overview of that particular case study. With that, we'll move to a couple of questions I have here coming through for Isaac. Isaac, our first question is, were there any further developments from the clinical program after this set of studies with this particular compound? And can you share any of the work that might have come from that?
Yes, after this study, which were bolus injections, we started with a series of infusions, slow infusions, faster infusions, And also, one of the side effects we saw with this, with the first studies was involuntary movements, which were moderate, not severely. And to do something about that, we had a couple of infusion studies where we also gave midazolam, which is a mild benzodiazepine, to shorten the involuntary movements. And of course, this is an anesthetic, which means it's not pain. It's not a painkiller. So we started doing studies with infusions and an infusion of fentanyl, which is an opioid against against pain. And after that, after a full set of studies, we started with patients with colonoscopy, and a couple of successful patients were tried out in another hospital, a nearby hospital, the Martini Hospital, in collaboration, of course, with how do you call them? Internal medicine specialist. But unfortunately, the company decided to focus on other things, and that was the end of this compound. The disadvantage of this compound was actually the very short duration. So in the labs of Doug Rains, they're thinking about analogs which have a slightly longer duration of action.
Wonderful, thank you, Isaac. And actually, we've got a question come in, I think back to Peter here, on, would we be able to explain the reduction in cytokine levels in treated subjects? And so one of the follow-up questions is, what was the route of administration as well for that?
This for Peter.
I think I misunderstood the question. Can you briefly repeat?
Yeah, so would you explain the reduction in cytokine levels in treated subjects? And then the route, they were curious about the route of administration.
Yeah, the route of administration of the compound of interest was oral. And the LPS, it's an IV bolus we administered. So, and the working hypothesis, how this, it's an IRAK1/4 inhibitor. So the preclinical pharmacology did show that there is effect on the various cytokines, also demonstrated in the LPS preclinical work. So we had good information in hand to mimic that in human beings.
Fantastic. Thank you for that, Peter. We'll move over to a question that we had come through for Marijke.
Yes, thank you.
Marijke, one of the questions was around any pushback that we might have received from the ethics committee in terms of performing that study. And if we had any, that was in terms of the bone marrow biopsy study, how did QPS overcome that?
Any pushback from the ethical committee? No, there was not so much pushback at the start of the study. I think the rationale, I think that's one important thing, that the rationale for why doing this study, performing this study in healthy volunteers and not in patients, I think that was very, written out and very clearly stated in a protocol, so that they didn't have any pushback there. I think at some point the sponsor would like to increase the dose, and then the ethical committee really requested to have a very solid rationale for why would they increase the dose, and also have clear projections on what would be then the exposure in healthy volunteers, and whether that was still safe or not. So I think that there was, maybe, I wouldn't say call it pushback, but there were questions for more data or more information. Yeah, but otherwise, No, and this is actually maybe also to allude to the point that Peter made with regards to the IRB that we're working with. They are, they're thinking very experienced, and they see a lot of quite complex trials in the whole space of early phase development. So I think in that sense there was, yeah, I wouldn't say they were used to it, but I think they, yeah, they have a good set of experts there to review these type of trials.
Wonderful, Margarita. And maybe the last question for you is, you know, how does one decide that an IMP could be studied in healthy volunteers, for example, similar to the study that you described here?
Yeah, you mean so so why not study it in patients? And how do you decide if you were already started in patients? How do you decide to go to healthy volunteers? So I think there were two things playing a role here. Of course, one, can we dose this IMP in healthy volunteers? So that's something you need to review and look at. And of course, there's several things that play a role then. So of course, you have your set of required genetic toxicity studies that you need to do. So you don't want to see any cytotoxicity of your compound. I think also what is important is the dose that you're aiming to test in healthy volunteers. And of course, also the prior experience you may already have with patients. And I mean, what is the safety profile you see there, and would you feel it's safe to bring that to healthy volunteers? So I think there are several components there. And of course, the other part is doing the bone marrow biopsies in healthy volunteers. Is that considered an acceptable risk? Yeah, and in discussion with the hematologist, they really thought that it would be if there's no possibility for studying this in patients, because they're in this case, there were oncology patients, so very fragile, and yeah, you don't want to go there with these patients, but in healthy volunteer, it would be acceptable. Yeah.
Wonderful. Thank you for that, Margarita. We've got a couple more coming in for Peter, so if we'll take it back to Peter, I think.
Yes, let's do that.
And this one is, what is your view on low-dose immune activators in healthy volunteers? For example, do you see low-dose CD40 agonist being feasible?
Yeah, a difficult question. It depends on, let's say, the preclinical profile and what is known in general. So this is typically, we normally when such a question comes through, we sit together with the specialist, medical specialist, eventually toxicologist, and so on to review the data. The same is true for let's say, oncolytic compounds. Same story. Can you dose them to healthy volunteers? Low dose, high dose, it depends on let's say, several aspects. So we treat them case by case. That's basically what our path forward is. So that is my, let's say, the view we take into account here at QPS. So there is no general answer, yes or no, but let's do it case by case, and depending on the profile of the compound.
Wonderful. Thank you for that, Peter. Well, I would like to thank each of you for the time and taking us through these complex case studies, and also really would like to thank the audience for taking the time to listen in on these. If you have further questions or questions that come in that we, for some reason, did not get to, please know that we'll follow up via email. Appreciate your attention. Also, I would just say if there's anything else that you need, you can clearly reach us at info@qps.com or go to our website for any additional information. Otherwise, thank you again for the time. Thank you to our scientific team for taking us through these case studies, and we look forward to next steps at QPS.